Hello everyone, good morning all of you. This is Dr. Sudarshan Sambhaji Pedge, Assistant Professor Department of Zoology, Kalasasi Rasika Mahadevani. Yesterday we have completed the introductory part of the metabolism of lipid. Okay, after the metabolism of lipid introductory part, we have seen today the beta oxidation pathway. So let's get started today's lecture. So first of all, what is meant by the beta oxidation and what will be the reaction it will be occurred at the time of beta oxidation. Okay, beta oxidation cycle is based on the beta oxidation theory proposed by the Franz Knupp. Okay, and the oxidation of the fatty acid occurs the beta carbon atom to form a beta keto acid. Okay, and the end carbon atoms at the carboxyl end are eliminated as acetyl coenzyme, the formation of a new carboxylic group at the site of the keto group. Okay, that is the one particular reaction is called the oxidation reaction. Okay, so you have see here the one particular cycle is here in the slide. Okay, so in that slide you have seen here there are the various types of reaction it will be occur here, and that would be the formation of the acetyl coenzyme. Okay, in the with the fatty acid it will be converted into the acetyl coenzyme, okay, in the last reaction. So this is the one particular in a cyclic form, that's why we call it as one particular pathway as well as the cycle. Okay, so we have seen all these reaction thoroughly one by one. So first reaction is the activation of fatty acid. You have seen here the saturated free fatty acid first undergoes the activation, okay, before entering the mitochondria. Okay, you have seen the fatty acid, if it will be the activated before the entering of the mitochondria. Okay, in the presence of ATP and ATP as well as the magnesium ion and coenzyme, the enzyme thiokinase, okay, or acyl coenzyme synthesis, okay, catalyze the conversion of free fatty acid to an active fatty acid acyl coenzyme. Okay, formation of acyl coenzyme. Okay, fatty acid it will be converted with the help of magnesium ion and thiokinase enzyme and formation of acyl coenzyme. Okay, so this is the first reaction. Another next is the second reaction. It will be the transport of acyl coenzyme into the mitochondrial matrix. Okay, you have seen here at the outer surface of mitochondrial membrane one particular acyl coenzyme cannot readily the transfer the inner mitochondrial membrane. Okay, a special transport mechanism is needed. Okay, and the transport is done by carnitine. Okay, you have seen in the carnitine one particular transport of membrane transport of acyl coenzyme, okay, in the mitochondria. So, okay, at the outer surface of the mitochondrial membrane, carnitine combines with the acyl coenzyme and forms acyl carnitine, okay, and the leaving the coenzyme at the cytoplasm. Okay, so the acyl carnitine then passes through the membrane, and at the inner surface of the membrane the acyl part is transferred into the intramitochondrial coenzyme, okay, to form a particular acyl coenzyme. Okay, so this is the one particular second reaction. You have seen here the acyl coenzyme, the help of carnitine, and it will be the enter into the mitochondrial membrane, after that the cytoplasm as well as the mitochondrial matrix. Okay, next is the third reaction. You have seen here the dehydration it will be occur, sorry, dehydrogenation it will be occur. Okay, so the acyl coenzyme that has entered the mitochondrial matrix undergoes the dehydrogenation in the alpha beta position. Okay, So resulting in the formation of alpha beta unsaturated acyl coenzyme. This reaction is catalyzed by the enzyme acyl coenzyme. Okay, so these dehydrogenase in the presence of the coenzyme or the flavoprotein you have seen here, which is which contains the FAD is the prosthetic group. Okay, and FAD is it will be converted into the FADH2. Okay, that is the dehydrogenase with the help of dehydrogenase enzyme. Okay, and the formation of alpha beta unsaturated acyl coenzyme it will be formed. Okay, so this is the dehydrogenation uh, reaction, third reaction. And next is the fourth reaction. This alpha beta unsaturated acyl coenzyme it will be hydration, it will be occur here. Okay, hydration means the addition of the water molecule. Okay, you have seen in the in this reaction. Okay, so in that alpha beta unsaturated acyl coenzyme after the hydration or the it will be the use of hydratase enzyme, they have converted into the beta hydroxyacyl coenzyme. Okay, so this is the one particular fourth reaction is the hydration reaction. After that number fifth is the second dehydrogenation. You have seen the beta hydroxyl acyl coenzyme here undergoes the dehydrogenation at the beta carbon, and hence the hydroxyl group at the beta position is converted to a keto group, thus forming a beta keto acyl coenzyme. Okay, coenzyme, that is the one particular with the help of beta hydroxyacyl coenzyme dehydrogenase enzyme. Okay, and at that time the NAD it will be the dehydrogenase in the presence of the NAD. The reduced coenzyme NADH is reoxidized by the respiratory chain. This is the one particular fifth reaction. After that, the final step is the cleavage. Okay, the final step in the cycle is the cleavage of beta ketoacyl coenzyme at the beta position in the presence of coenzyme. Okay, this is the one particular reaction, or that reaction is called the cleavage or the thiolysis. Okay, or that cleavage is called as thiolysis, and the enzyme that catalyzes the thiolysis is known as the beta keto thiolase enzyme. Okay, beta keto thiolase enzyme. As a result, a molecule of acetyl coenzyme and acyl coenzyme are formed, of which the acetyl coenzyme. Okay, so this is the detoxidation process. Okay, thank you very much.